For those of you who don't know me, I am the other John Hoffman in the MTO office, just to make things interesting. I am, the vast majority of my portfolio is within the organics circuits component of Whitney's office strategy. But today I'm going to talk about two topics that are actually focused in the microsystems manufacturing ecosystem portion of her vision. I am fully embracing Whitney's vision of new and interesting ways of manufacturing materials and microsystems in ways that we can't do it today. And in support of that, then, to manufacture the new photonic, quantum, and organic technologies of tomorrow. So my talk today is going to be broken into two parts. The first part of the talk is going to be focused on a new method for manufacturing new materials that don't currently exist in nature. And the next portion of my talk will be using these types of materials to fabricate the microsystems of tomorrow. Okay, so the first part of my talk is going to be focused on sequence-defined polymers and the application spaces that they can be used in. So for those of you who aren't familiar with the sequence-defined polymer, I'd like to direct your attention to the pearl necklace in the center of the image, of the slide. And so in there you see a string of what I'll call monomers, and in a sequence-defined polymer, the identity of that monomer and its stereochemistry is controlled. So as we move from position to position down that string, we have complete control over the properties of that monomer. So why are sequence-defined polymers interesting? You can think into the biological space. There are molecules like proteins that are sequence-defined polymers, and they can fold into these really amazing shapes that give them unheard of types of function. What we'd like to do is extend that space away from molecules that can only form these structures and do these functions in water. Can we think of sequence-defined polymers that can form interesting three-dimensional shapes and do interesting functions outside of an aqueous environment? So if we can do that, moving over to the left-hand side of the screen, looking at applications, we think we can use these materials in microsystems manufacturing to give devices properties and capabilities that you can't see today. But we also believe that if we can do this for microsystems, we can enable a whole host of other application spaces. And what that's going to require is a platform manufacturing capability where we can sequence almost any type of polymer with any backbone chemistry, with any type of monomer, and do that kind of a plug and play on the fly capability. So when we look today at sequence-defined polymers, what are the limitations that prevent us from synthesizing these molecules? Every time we see a new polymer and we have some new idea for its application, we need to use unique reagents. Every synthesis process is unique. And because of the errors that are associated in these synthesis processes, that you get the polydispersed mixture of molecules. And so you need to go through very unique purification steps for each type of polymer that you make. That's extremely challenging, and we're not very good at it today. And so because of that, as we try to move from creating these sequence-defined polymers and think about their applications, it's very difficult for us to predict how will these molecules, if we can even make them, how will they fold into secondary structures, and then potentially the sorts of functions that they could enable, such as self-assembly. So if we look a little bit closer at the techniques that are available today to synthesize these molecules, one of the first places to look is to nature's machines. The image in the top left is a ribosome. I believe my colleague Juanan introduced these earlier today. For those of you who aren't familiar with a ribosome, the ribosome is a molecular machine that uses a template and uses that template to write a precision sequence-defined polymer. There are many of these types of machines in nature, and some of their attributes are that they're extremely high throughput. They write these polymers with extremely low error rates, and they can write polymers over a broad range of lengths, from just a few monomers all the way up to many tens or potentially hundreds of thousands. The limitation, though, with these systems is that the chemical variety in which they can synthesize is relatively small. In the case of the ribosome, you can only use a handful of monomers, 20 of them. These are the naturally occurring amino acids. And then there's only a single backbone chemistry that is enabled, and those are amide bonds. So if we think of nature's machines and their attributes, the inverse is what we find in synthetic chemistry. In synthetic chemistry, what I'm showing here is an example of iterative step growth. These polymers, when synthesized, have attributes that they're not very high throughput. You can only add a few monomers in any given time. The error rate is extremely high, meaning you get these very polydispersed samples that I talked about before. And the length range that you can synthesize over is very narrow. The upside, though, of these systems is that there's an extremely wide chemical variety, and I think we can see that in all the synthetic polymers that we're able to synthesize today. So is there a way that we can bring the best of these two different types of systems and bring them together? So one way that I'm thinking about this, and this is depicted on the right-hand portion of the slide, is to use what's called molecular engineering. And molecular engineering is a design, build, test, learn process that's composed of two major elements. The first, depicted by the computer, is computational molecular design. And on the right-hand side, the Petri dish is our processes for the build, test, and learn portions of the cycle. You can think of directed evolution as one of those types of processes. So this capability of molecular engineering has enabled two very, very important discoveries, in my opinion. The first, depicted on the left-hand slide, this process has been able to create molecules, proteins, that can synthesize chemistries, using chemistries that are new not only to nature, but in many cases new to chemistry. There are certain chemical reactions that can only be done by these designed proteins. On the other half of this figure, there is a picture of a structure where very deliberately different proteins are brought together into very well-defined three-dimensional space. I'm thinking, is there a way for us to combine these two things as a platform manufacturing technology? Can we plug and play each of these proteins with these unique chemical synthesis properties into three-dimensional structures that allow us to define and synthesize sequence-controlled polymers? I think this is a very unique approach and just one way to do it, but I'm very interested in speaking with you and working with you to develop new ways of writing these polymers. So this is the last slide in this section. Next, I'm going to move on to ideas for how we can use these types of materials to build the microsystems of the future. So when we look today at current synthesis, manufacturing technologies, we'll start over on the left-hand side and looking at the channel materials and the patterning processes that are available to us. In these cases, we are limited to two-dimensional patterns that we can write on surfaces. The example I'm showing here is a substrate. It could be silicon. You're growing another inorganic material on top of it, and you're limited by the planar lithography in the types of geometries that can be developed. Can we imagine building channel materials and different constructs for these devices in true 3 dimensions? And that's currently not available to us with manufacturing technologies. On the right-hand side is chip integration. And here also we're limited by the geometries that we can realize. While we're able to manufacture devices and systems with extremely powerful capabilities, we are limited by either stacking chips right on top of each other or separating them through a substrate. Can we think of other ways of integrating these materials in 3 dimensions to realize devices that we just can't realize today, and potentially capabilities that we can't realize through these technologies? So looking at biopolymers as one particular type of sequence-defined polymer, we get some inspiration that we could potentially overcome some of these limitations and realize these devices. On the left-hand side of the image is a nanometer scale templated growth. On the left is dictated by a protein system, which can nucleate zinc oxide in very precise locations. And on the right is a DNA origami-based synthesis technique where they're templating the synthesis of gold structures. This gives me inspiration that we could use these sequence-defined polymers to grow materials in arbitrary three-dimensional shapes and create entirely new types of device architectures. Moving over to the right-hand side, we can use these same self-assembly techniques to pattern materials that generate entirely new types of device integration. Here I'm envisioning, opening up the vertical side of these devices and be able to create entirely new forms of interconnects that can overcome some of the bandwidth issues and heat issues and others that we saw with traditional heterogeneous integration. It's my feeling that if we can demonstrate some of these first approaches to this and opening up this vertical plane, that we could eventually realize true three-dimensional microsystem architectures, where we're bringing together disparate materials, disparate functions, all together into a single device. So that's it for my talk. I very much like to work with you on both of these two topic areas. And I look forward to talking with you outside afterwards and seeing what we can create together. Thank you very much.